Okay, hello everyone. We see each other again. And today I'm going to present the chapter 8. Oh my god, I even forgot to let me share my screen. How can I forgot to do that? So sorry. Maybe just the end of the spring break. So today I'm going to present the chapter 8, and it's talking about the vertebrate opsins. So most of there are about like 9 different types of opsins genes in human, and generally can be separated into different subgroup depending on activated G proteins, what kind of G protein, GI or GS or GQ. And of course, today we'll go through like generally like some options that's very traditional. But today we are going to focus on those non-canonical opsins like peropsin, peropsin, and long wavelength stimulate opsin from the butterfly. Rhodopsin families is a huge. Most of them are like chloride pump, proton pump, and some channels, and also enzymes that activate adenylene cyclase (AC) or PDE. And in vertebrate, most of them are coupled with G protein. It's functional as a G protein coupled receptors. So for G protein coupled receptors, they are like downstreams partners like GS, GI, GQ, and G12, G13, and their functions they have like and change the concentration of different secondary message like cyclin AMP, calcium, and some may even change some proteins, and it will affect the gene expressions in the nucleus. So in this book, generally say that there are about 8 types of different opsins. Some activate GT, and today one of the opsins that we're going to talk about the Parapinopsins that's coming from the lamprey or other fish, first discovered in catfish. And this one is a GT coupled, and other GI or GO coupled or GS coupled. And we know that melanopsin is a GQ coupled. It has a very long time course, and also the butterfly long wave length sensitive the opsin is also GQ coupled. And also neuropsin. And today we're also going to talking about the peropsin. It's a spider peropsin. And it's very unique because it's a functional in the dark, but it's like inactivate by the light. And also RGR is a retinal photoisomerization, isomerize. And actually, let me see, peropsin is quite like a peropsin. Yeah, peropsin actually is quite also like a photoisomerize because expression in the mouse retina, retina pigmental epithelium, it binding to the ultras retina and can change the conformation to the nemesis retina. So it's also kind of function as a photoisomerizes. So for human, and this is human opsin, totally 9. And very traditional is like the 3 types of opsin: red, green, and blue opsin, and rhodopsin. They all function is a region. Melanopsin is retinal ganglion cells and related with the circadian function. And all of these others we're going to talking about today. It's the Parapinopsin, peropsin, and long wavelength sensitive bistable opsin. So each of them is very unique. So for for other traditional opsins in the vertebrate or in human, we can say it's that in the dark state is binding to 11-cis retina after capture photon, and it will change the conformation to the ultra's retina, and the opsins will become changed to another conformation, another state, and then chromophore will be dissociated with the protein part of the pigment, and the whole pigment becomes inactivated. But there's also bistable pigment that exists in the natural world. That's the bleaching resistant pigment. That means that after capture photon, although it will also change like from the 11-cis retina to ultra's retina, but it will not dissociate from the protein. So with another light wavelength, usually it's a longer light wavelength, it can go back to change back to the dark state, to the rest state. So, and this is a quite stable, and the chromophore do not dissociate, so there's no bleaching. So first, we're going to talking about the Parapinopsin. Parapinopsins were found in the lamprey, in the frog, or the other vertebrate. It's first identified in catfish. It's located just on the center of the top of the head. And this part is a non-pigment pinot window. So there's no pigment cells here. So light can penetrate the skin and the skull, the bones, and be captured by the receptors that are located and just at the top of the fish's head. And the GT coupled on UV light sensitive pigment. It activated GI, but of course, after molecular engineering, it can change to uncoupling to other G proteins. An important part is that it's a bistable opsin, so it can capture with different light. So we can see here as a bistable opsin. So at the dark state, they do not absorb any light, but after activate with UV light, and most of the opsins will prefer to absorb like a green light stimulation. Then after absorbing after irradiated with the UV light, then the next step, if irradiated with the green light, then absorption way curve will indicate that most of this pigment will be absorbing a UV light that wavelength. That means that after providing the green light, and the pigments will go back to switch to and still rising state and dark state. Let's go back to here. So this image is showing in this functional. Since it's coupled with a GI, so after activate with the UV light, you will decrease the concentration of cyclin GMP. So this experiment is done on photoreceptor expressing the calcium sensor. So this result is indicate the calcium level, that's intracellular calcium level. But we know that because in photoreceptors it's a CNG channel that gated gated this calcium influx. Sorry, it's cyclin AMP gated CNG channel. So using this fluoxetine treatment, and you will activate adenine cyclase and will huge increase of the cyclin AMP. And so as a result of increasing of the cyclin AMP, the calcium level will also be increased. Then shedding with this UV light, since this opsin is coupled with a GI, so it will decrease the concentration of cyclin AMP. So as a consequence, the calcium concentration will also be decreased. And at this stage, and there's a shedding more light, this shows no difference. But after provide a green light, and some of the pigments will go back to a dark state. So that means that in an inactive state, and it not activate the GI signal pathway, and the calcium concentration will continues go high until shedding another time another light pulse of the UV light, such the GI signal will be activated again. So all of this shows that this work because it's a photoreceptor cells, and works that these signals can be like expression and in another species. It can be controlled by light. And as a control, here it shows normal opsins that from golden fish. And this opsin that shows that will cause all the decrease of this of the calcium signal, and it will not be changed by with another wavelength, the green light. So it means that for the goldfish opsin is not bistable. However, for this Parapinopsin, it's a bistable and can be recovered by another a different wavelength photons. So it's shown here because it activated the GI. So when it's shown here, after shedding with the UV light, it will go to this photoproduct, and you will activate the GI signal pathway, and it can be turned off and after shedding this green light. So here it shows the molecular structure of this Parapinopsin, and show that it can couple with a G protein. This is definitely is a G proteins. It shows that this is a retinol. So here it shows another experiment setting, show that so after shading the UV light, it has a very long response, and this response can be stopped by shading with a blue light at a 470 nanometer light, and this will be shut down. So this is a very typical experiment setting to demonstrate that this is a bistable opsin. Then let's move on to take a look at the peropsin. Peropsin is in another species that's an illicit. So this creature is very unique. It's an animal, but you will insert into the sand bedding of like the lake. So or it's like the movement like a fin or is like degenerated, and the muscles were formed myotome and surrounding the spines. And it's a long term. It seems that there's no brain, but actually at the tip of this at one end of the spine, they still show some very like very simple structure that can be determined. Can call them like a hindbrain or something, but the whole animal is really like enlisted, and seems like its journey is controlled by a spine that lines from head to the tail. So from this animal, scientists isolate a very unique opsin, that's the peropsin. So later in the mouse eyes, peropsin is also been discovered that it's located in the retinal pigmental epithelium cells, and that's very close to the membrane of this epithelium cells. So it's functional as a retinal photoisomerase, and it contains the DRY motif, and which is high conserved among GPCR, but in so far as no experimental provements show that in the retinal pigment epithelium cells, if this peropsin really function to activate the G protein signaling, but it has the motif that can binding with a G protein. But the interesting part is that it's not functional at the light activate state. So after activate, like it will change because it's binding. The difference is that it's binding in the retinal state. It's not binding in the evidences retina. It's binding on ultras retina. On the ultras retina, after shedding with the light, it will change the conformation of the ultras retina to the evidences retina, but it's inactivated. It will not activate a G protein. However, in a dark state, it can drive the G protein signaling while it's binding the ultras retina. And the absorbing of the light will inactivate the peropsin. So that's a totally different. So for most of other G proteins, opsins, after capturing light, it becomes active, and it will binding and activate the G protein signal cascade. But for peropsin, it's just the opposite. In the dark state, it's binding ultras retina, and it can have an activate the GPCR function. That's quite unique. So here we see that in the dark state, it indeed have some absorptions, and also that absorption is different from the light state. In the dark state, you can activate the G protein until it's being inactivated by the light. Of course, it's also more binding with the ultras retina. So this glial sensor CMP assay, in this assay, it shows that after shedding the light, it shows us a CMP concentration. So compared with a different opsin, with peropsin, So this is peropsin. This is a peropsin. It's coming from spider, and makes some necessary mutations that it becomes a GI coupled opsin. So we can see here, after shading with the activated light, the cyclin P will be decreased in this GI coupled opsin. It will have no effect with the original one, and the jellyfish option will have an increase. Maybe it's binding to GQ or GT. And also we can see, compared to see the mosquito opsin, opsin 3, it's binding to a GI. So we can see that after shedding light, the cyclin P's concentration was decreased. And this, after shedding light, its cyclin P concentration is increased. So now let's move on to see the long wavelength sensitive bistable opsin. That's LWS opsin. It's isolated from a butterfly. So for a human being, the color information is coming from the short wavelengths, medium wavelengths, and long wavelengths. But for butterfly, it's a very vivid colored world. It has 5 opsins that can sensitive to UV light, violet light, narrow blue, single green, and red. And for this long wavelength, its peak the optimal simulation wavelength is a quite a long wavelength. That's a 575 nanometer. So I have to do some like molecular engineering that can change the third transmembrane domain, and sorry, it's not transdermal domain, it's the third cytoplasmal loop region, and that will be just switch into a GS coupled opsin. So because it's So now let's summarize all these canonical opsins, like the isolated from the catfish, the Parapinopsins. The most critical part or the take-home message is that for the Parapinopsin, it's a bistable. It's a bistable opsin. And for the peropsins, we need to remember that it's activated spontaneously in the dark, being activated by light. And for the long wavelength sensitive opsin from the butterfly, of course, it's the long wavelength. So this opsin gene is the longest spectrum sensitive. So far, we know that among all optogenetics toolbox. So here is the reference that was cited today, and thank you.